Thank you, Faraz. Appreciate that nice introduction. So
Mhm.
thank you for everyone for giving me the opportunity to present today. Faraz did a really good job of to talk about my title, so we'll get right into that. So this is just a normal Merck disclaimer, so I'm not going to go through that. It's an eye reading process. So again, Faraz did a really good job. I've been managing the department here within drug product development and new technologies for more than 27 years. My roles have been across multiple programs, a lot of live virus experiences. That includes Ervebo, as he just alluded to, Procod, Varivax, and Zostavax. But my team has also been really heavily engaged in some of these new technologies that we're going to talk about today that are really going to enable the vaccines of our future. This includes lyospheres, microwave vacuum drying, ID and MAP technologies, and alternative delivery. And if you ever do run into me in an actual conference when we're face to face again, some conversation starters, I've had the opportunity to coach with an NFL head coach. I'm active in high school coaching as well as youth sports. I love tequila, so anybody wants to sit down and talk about that, please come and find me. And I've also eaten in the oldest restaurant in continuous operation, which is Butun in Madrid, Spain. Really highly recommend it if you ever get a chance to have some. So I really want to talk about three things today. First, what Merck's vision is about opportunities in the drug product development space and the vision that we're focusing on moving forward. Some of that will lead to some of our new drying technologies that we're championing within Merck. This includes lyospheres and microwave vacuum drying, as well as looking at alternative delivery technologies and what innovations can really help set the tone for increasing vaccine coverage across the world. This includes intradermal delivery, microwave patch technologies, as well as sustained controlled pulsatile release. So Merck has a long history of innovation within vaccines as well as drug product development. This dating back to when Dr. Maurice Hilleman was part of our company. We've been at this for more than 100 years, and we've been able to bring in more than 40 vaccines into human health as well as animal health mindsets. And when you look at the history of Merck's vaccines, starting all the way back in the 1960s with licensing in the first measles vaccine in 1963, upwards to today of Ervebo and our recently Vaccinuvance, which is the PCV15 vaccine that we recently have achieved licensure for. So why are vaccines an attractive market? And they really do have a huge impact on significant public health. And I just articulated two here. So if you look back in 1941, over 890,000 people contracted measles. When you look at back in 2010, only 63 people actually contracted measles in 2010. There were more people that went to the summit of Mount Everest in a single day, 169, than those that had contracted measles. Looking in 1900, 48,000 had smallpox. 1921, 207,000 had diphtheria, and 21,000 in 1952 contracted polio, compared to in 2012, where 0 cases of smallpox, diphtheria, or polio have ever occurred. This really shows the public health benefit. And we all know what we're ongoing through right now with COVID and the importance of vaccines and helping us get through this global pandemic. So there is a dramatic public health and economic impact. We can save over 9 million lives annually by giving vaccines. And there's $295 billion in direct US costs saved by using vaccines within the US alone. It is a large and attractive market with a 30 billion target in 2015, growing globally to 50 billion by 2025. We have durable revenues and unique return on investments. We have fewer competitors. We do have a higher barrier to enter into this space. We do have a low patent cliff and a longer lifespan of these programs overall. So if that's the case, why are not more people involved in vaccine drug product development? And one of them is the complexity of vaccines. When you look at the size of them, as well as the complexity, they continue to increase over time relative to a small molecule. You have to deal with protein modifications, such as aggregation, hydrolysis, deamidation, oxidation, and cleavage. You have to worry about the viral envelope in the case of a live virus. And what happens if you lose that envelope? What's its composition? How fluid is that live virus's envelope? And you also have to look at new nucleic acid damage, oxidation, depurination, and degradation. And within the messenger RNA space, we all know the importance of keeping that messenger RNA stable so they can produce the protein that we want. So within Merck, what is our expanding our vaccine coverage worldwide and our strategy looking at? We really are looking to want to protect a broader population and future generations of preventable and treatable diseases. Our goal is to double the revenue in the next 10 years and quadruple the number of people that are being protected. How is Merck going about this? We're looking at our pipeline and how we can accelerate that faster. You're looking at a supply chain and how you can improve global access of these vaccines. And then, of course, you have the customer developing the global approach to the business and giving them the ability to have consumer-friendly products. So we're looking at targeted new infectious disease targets, leveraging open innovation, which I'll talk about in a minute, and developing novel delivery and manufacturing approaches. From a supply chain perspective, we're looking at what do we need to have global product profiles, thermal stability, and improving the cold chain storage of our vaccines, and enabling low-cost, high-volume manufacturing. From a customer perspective, how do we get into the core penetration more? How do we stabilize and expand our markets and supply? Lower those manufacturing costs again, and improve that customer convenience. And we'll talk a little bit about that as we go through some of these alternatives. So within my area and the new technologies exploration, we really want to advance innovation opportunities that help drive these business goals. This includes improving the affordability and accessibility of our products. And there's really two types of innovation we've been looking at. Closed innovation, where that is something that within Merck, we have the expertise and the know-how to do, as well as open innovation, where we have to go outside of Merck's walls to figure out who we can partner with and champion these technologies. And some of these we'll talk about today. Lyosphere technology is an internal investment. We're looking at microwave vacuum drying, particle delivery, implants, Vaxis nanopatch, and nanopass microjet. These allow us to look at thermal stabilization, drying technologies, skin delivery, alternative delivery, controlled pulsatile release, and modern adjuvants. The process, the product, and the customer are key focus across these different innovations. So today, most of us know that we put our materials in a standard vial, blow-field seal, and we lyophilize. But the challenges with that remain. Drug product manufacturing is capital intensive, usually has long cycle times. We have sometimes overusage and inefficient usage of our drug substance, which impacts our supply. We also have to worry about cold chain distribution and how we get it to where we need to get it to, right? Moving into the future, our gold standard has always been to lyophilize in a glass vial. But to be able to meet the needs for public health, we need to think about new innovative ways of drying our products, improve our product stability, lower our costs, provide flexible production capacity, and improve drug product device combinations and facilitate novel product combinations. And one technology that I'm going to speak upon now is Lyosphere technology. This could really provide a solution to a lot of these issues as we move into the future. So most of you know this is the standard way we lyophilize our products today. Here's a large lyophilization cabinet being loaded with a multitudes of vials. Is there a better alternative to this? And we think within Merck, yes, there is. So luckily within Merck, there's a technology that our animal health colleagues champion called Sphereons, which has been used to make oral poultry vaccines for quite some time. What we've been able to do is meet with them to talk about this technology and could we apply it to human health products? And the answer we believe is yes. So up here at the top, you see some of our developmental labs that we have within our space to look at this technology. And what it can be amenable to is different images for different markets and the supply chain flexibility that we may need. So what is Lyospheres and how does the process work? So think about making your standard formulation just like you would normally do. You would then take that formulation and you freeze it into tiny little beads on a very cold plate. Those beads are then shuffled into a bulk tray, lyophilized in bulk, and then being able to be dispensed into any primary package that you may want. So again, looking at them, they look like tiny little spheres, right? We can vary the size of those spheres from 10 microliters up to 500 microliters in size, giving us the ability to vary that dose that may be needed. It is dried in bulk relative to single dose vials, and allows you to have dose titration ability, what you can do up here, and be able to change and mix and match products that you may want to put together that may not have been able to be co-formulated previously. We have shown in our development, and I'll talk to that in some of our data, that this is amenable to live viruses, antibodies, therapeutic proteins, and small molecules. We have shown that through this technology, we can actually improve our thermal stability characteristics. Again, I'll touch upon that soon with both the monoclonal and a live virus example, and allows us to have right first time processing. So we don't have to worry about drug substance losses during processing, because I can actually dose titrate off of my bead potencies, vary the amount of beads that go into my final drug product, and is amenable to many different images. I don't have to change my process to fill into all these different images. All I have to do is count my beads into whatever image today is needed, whether it be a single dose vial, a multi-dose vial, pre-filled syringe, cup. We can do that by using this technology. So up here are a couple of videos of what it really looks like as it's going through. This is our development machine being able to make the beads on the frozen plate. This video sort of shows the plate being actually, these beads being shuffled out. They are collected into a standard bulk container. Those beads are then able to be dispensed into any image we want. And this is our development line where we're using this to be able to dispense these beads. And those can go into whatever image that we may want to happen. So this is sort of like what our development units look like. So I've shown you a little bit of the technology. What about how the technology actually works, and do we actually get these goals? So here on the left, you're seeing some data from one of our live virus programs within our network. And historically, at 1 week at 37 degrees stability, which is that VBM7 requirement to get into some of those thermally stable situations, we were seeing by standard lyophilization a 1 to 2 log loss over potency. By allowing us to up the high disaccharide levels in the formulation to 25% sucrose or 25% trehalose, we were able to take and knock this loss rate down almost in half, improving the success of being able to have a thermally stable program on a live virus. In addition to that, we've actually explored monoclonal antibodies and getting to a room temperature product. Here, what you're seeing is the standard lyophilization formulation, which would require us to have 5% aggregation state after 6 months at 40 degrees stable. We have taken a high disaccharide formulation again and being able to show that we can stabilize this material out to 6 months at 40 degrees with a very small change in aggregation state. Again, allowing us to potentially get to a room temperature stable product that was not amenable by standard lyophilization. So we haven't stopped there. We've taken this technology and said, could we use this technology to enterically coat our lyospheres and be able to deliver to a specific part of the gut? Again, the goal is to optimize that formulation, dry it as we normally would, identifying a coating process that we could actually coat these lyospheres to allow them to get through your GI tract and into your small intestines. And we really wanted to be able to say, could we do this technology or not? And the answer is yes. We've been able to take a model protein, myoglobin. We did not have an impact to yield or structure when we went through this process of coating and lyophilizing and drying in a lyosphere. And we were able then to also be able to dissolute it into the proper part of the gut, showing that we could actually deliver it to the small intestines if we wanted to go there. This is another aspect of this technology that we're exploring. So I hope I've been able to show you at least quickly through lyospheres some of the benefits that it can give us. It allows us to have new mix and match approaches, so new differentiation of products. It allows us to have liquid-dry combinations. It allows us to have a lower cost and a smaller cold chain footprint because I can improve my thermal stability. It allows me to have different images that I could potentially use this product for. On the manufacturing side gains, we can minimize our overages because we're dose titrating the actual dose of each bead, so I can fill the right amount of beads into my final image. It lowers my drug substance waste because I have right first time technology here that allows me to dose titrate to ensure I have the right dose each and every time. I can increase my throughput of lyo cabinets up to 2 to 4-fold that we have actually proven. And this has all been proven with our animal health colleagues in Salamanca, and we are well on our way to having a sterile lyosphere facility within Merck to be able to make these for clinical materials in the next 2 to 3 years. So that's one of our drying technologies we're looking at about. The other is microwave vacuum drying. And again, this technology is coming out of our collaborations with Enwave and what they've done in the food industry, which I can dry products instead of in days, within hours. And that'll be a huge savings to us in the long run. It is a technology that applies a microwave under vacuum, which allows for rapid drying of the processes. What we have shown that this is a faster drying technology and is amenable to both semi-continuous and potentially continuous manufacturing. It is compatible with multiple images, whether it be a pre-filled syringe, a dual chamber, or making pellets. We have shown that we can use high disaccharide formulations similarly to what we have done with lyospheres and is amenable to VLPs, LBVs, and monoclonal antibodies. Its footprint in the grade A space is much less, and this can help lower the overall capital cost of operation of these technologies versus our current lyophilization process. And we've been able to demonstrate that we can dry materials in 8 to 12 hours relative to days in a standard lyophilization chamber. So here are some of the data that we have to show from a VLP that we have used. We've looked at lyo versus rev drying, which is the microwave vacuum drying. No change in Viacore potencies, no change in reconstitution times, variability in results, or in the moisture results. And here's a few different formulations that we've been able to put into our microwave vacuum drying and show to keep our potency across these consistent, whether we use cycle 1, 2, or 3. This is very promising to allow us to move in this direction. In addition, we've taken an LVV and done the same processes using some of these formulations, or a cycle that normally would take 45 hours or longer, we can dry in 7 hours. Similar potency results, same reconstitution times, and variability. And these are just some of the cakes that you can see the difference between microwave vacuum drying on the left and a lyophilized material on the right, showing that these high disaccharide formulations can be dried in hours instead of days without impacting potency in either a VLP or an LVV. Another example of this is looking at this, where we've been able to take Rev, a standard formulation containing 12.5% sucrose and 12.5% trehalose, 7-hour cycle time versus 168-hour cycle that was not optimized at this point. However, if you look at the stability data of these two formulations side by side, you can see the improvement in both 1 week and 2 week of thermal stability profile relative to drying the same things in a lyo cycle. A substantial improvement, which would improve our thermal stability profiles of the product. Similarly, on the point of a monoclonal, taking something at 50 mg per mil, we've been able to flatline the overall aggregation state by going to these high disaccharide levels relative to the standard 7% or lower saccharide levels seen in the current formulation. So taking this technology, this was a unit that we had in our development space up until 2018. We worked with Enwave to build and generate the first and only of its kind GMP unit of Rev drying, which is the second generation here. And I'm proud to say that we currently have this within our developmental space right now being utilized. It is both GMP and non-GMP for development purposes. We are going to take this unit and move it into our clinical facility to be able to make material using this technology in the not too distant future. So those touch upon some of our drying technologies. What are some of the other things that are important when we're talking about alternative delivery? And this is looking at either intranasal, oral, ID delivery versus the standard needle syringe. So why look at alternative delivery and specifically to the skin? Well, really, when you think about it, ID delivery can answer a lot of questions. It's cost neutral or cost savings, needle-free and pain-free in the high-income markets. From Merck, we're hoping that it becomes dose-sparing and decrease our cost of goods. In the low and income markets, it allows for low cost, increased stability potentially, to decrease the need of the cold chain, and ease of administration. So background on ID delivery, I think most may know of, it improves vaccines using a smaller needle. It can potentially be dose sparing that we've seen for varicella, flu, and rabies. And we'll talk a little bit about some of the data we have from our Zostavax trial that we have used this technology. It has improved immunogenicity compared to sub-Q or IM, and it is really an area of focus by WHO, UNICEF, BARDA, BMGF, PATH to be able to put measles rubella on a microwave patch into the future. There are different layers of skin and delivery differences between what you look at from a nano patch to a micron jet to the Soluvia device. And all these will have different impacts in your immunogenicity moving into the future. So a case study that we have done with Zostavax using the Micron Jet. So all this is taking this little nanopass device from Micron Jet's nanopass device, putting it on a needle syringe and injecting it. And the goal was to take Zostavax and look at whether or not using this technology, could we be dose sparing and get similar results. The clinical study involved 221 patients. They received either full dose or a third dose of the vaccine administered sub-Q with a needle syringe, or with the Micron Jet, you're looking at a full dose, a third, a 10th, and 127th. To look at the immunogenicity associated with it, we looked at two assays, either a GP ELISA for antivirus varicella antibodies or an ELISpot assay, which will give us memory responses. For the first one, 6 weeks to 18 months of post-vaccination, what we were able to observe is that ID at 127th, the dose of a full sub-Q, were very much comparable overall. When you look at the ELISpot assay, which is showing memory, again, showing that full dose versus 127th resulted in similar results in that material. So showing us that this technology could be amenable long-term. So one of the things that we have continued to work towards as a fact of this is looking at microarray patches and how this technology can be applied into the future. So up here is a video of a device from Vaxxus that allows you to put this material on and put that on your skin. The benefits of microarray patch, I think most of you know. There's simple application, single use, less painful. The challenges to this point are really that there is no vaccines by MAPS as we speak today. These do require you to wear it on your skin up to 30 to 2 minutes or longer when you're talking about dissolvable. But Merck has entered into an exclusive license to develop a high density microarray vaccine patch. We are planning to go into no-go to clinical preparation later in this year, and then have material into the clinical setting in 22 to 23 timeframe. We are also not only looking at a solid microarray patch, but we're also looking at dissolvable technologies. So on top of looking at ID delivery, what else is out there? Well, one of the others is drug delivery systems. And how can you give one shot or one visit to the doctor and be able to get not only your prime, but your boost? And one day to do that is looking at some type of sustained or pulsatile release. And why would we want to do this? Well, one, of course, is we want to increase vaccine coverage. And how you can go about it are two different ways. Sustained release, where you inject it once, and over time, the dose is slowly released. The second is to more of the conceptual mindset of a pulsatile release, where you're injected once, and over time, you get a release of your product multiple times, similar to what we do in a doctor's office today, where you come in at 0, 2, and 6 months and get your doses. Here you come in and get one. And to do this, are you thinking about some type of stable microspheric technology or microparticle that will allow you to inject once and get both your prime and boost in one dose? By doing this, we can expect to increase vaccine coverage so that you have fewer immunizations needed, which leads to greater coverage. So two examples of this technology that are out there today: Iowa State on the left using a polyanhydride vaccine concept, where you can change the ratio of these components and allow you to have different release kinetics of your materials. In addition, there's the seal technology that MIT has championed, which allows you to be able to do the similar thing using a PLGA type backdrop. Within Merck, we are exploring both these technologies to see whether or not we can advance this into the future. So I hope through this talk, I've been able to show where we are today. We are a form-fill expertise in our vaccines of LVBs, subunit, VLPs, conjugate, messenger RNA. We've explored adjuvants. We have non-GMP lyosphere and microwave vacuum drying equipment, and we are having multitudes of external alliances to explore. Within the next 5 years, we want to be able to expand this, implement sterile lyospheres for our inline as well as pipeline products. We are establishing connections and connectivity to advance ID delivery, whether that be pulsatile, controlled, sustained release. We are looking at new adjuvants to improve immunogenicity in those materials. And we are looking at self-administration of vaccines in the future on either solid or dissolvable microarray patches. These technologies we plan to move into the future with: lyospheres, microwave vacuum drying, various new images that allow us to have flexibility for that consumer. We have control of pulsatile release that can get you that prime boost together, and even novel adjuvants, which we haven't even touched upon today. as another way of improving our immunogenicity and getting more people vaccinated and covered effectively. So I hope through this summary, you can see where Merck is going, that we really want to enable world-class supply through flexible manufacturing and alternative drug delivery. We have two technologies from a drying perspective that we're pursuing: lyospheres, allowing for an agile manufacturing platform that can be applied to a broad spectrum of products for bulk drying, dispensing, and stockpiling; and microwave vacuum drying, which can allow us to have drying in hours instead of days, as well as operating in a semi-continuous to continuous mode. In addition, we are really thinking about that patient and that centric world of being able to look at ID delivery, where we can have patient convenience and compliance increased, potentially be dose sparing, and therefore lowering our cost of goods, reducing pain, and increased thermal stability profiles. I would be remiss with not thanking the team within my organization that have been helping to support these activities, as well as my leadership team, right? So these people have been heavily involved in bringing these technologies forward, and I look forward to any questions that any may have at this point. Thank you.